Okay. So, as I said, those who doesn't express the surface immunoglobulin, they die, and those who have very fitted surface immunoglobulin, finally they will survive, and the rest of the cells, as you can see here, they will die. The reason they die because they cannot get help from the T cells and dendritic cells. So those who have a fitted surface immunoglobulin, they get a signal from these two cells and finally become survive. And some of them go to produce IgG, become plasma cell, long-lived plasma cell, and some of them differentiated to memory cell. So you know how these cells is produced now. If you have a question now, I can answer it. Okay. Another thing which is important in the germinal center, it is isotype switching, because they have high affinity maturation, somatic maturation, and also isotype switching. We already known about two others, so now we're talking about the isotype switching and how it happened. So when the B cell activated, they get signal. You know that CD40 ligand, and also the cytokine. I said it is very important for activation. So we saw that CD40 ligand, how important it is. It is very important for generation of the germinal center and high affinity antibody. We already talked about that. We mentioned they have an isotype switching. Isotype switching is done by help of the cytokines. So when they are in the germinal center, based on what kind of cytokine the T cell is providing, they can produce either IgG, IgE, or IgA, based on the signal they receive from the T cells. Why T cell give different signal? Based on the type of the antigen they activated. So it's no wonder when it is intracellular bacteria, they usually produce interferon gamma. Why? Because the interferon gamma have to activate macrophage to kill them. But if it is extracellular or allergen, they produce interleukin-4, because they need to produce some IgE or some immunoglobulin to help the mast cells and eosinophil to kill them. So based on what kind of antigen is that, the T cell provide appropriate cytokine. So if they produce interferon gamma, they become IgG subclasses. If become IL-4, they become IgE mainly. So I always say mainly. It doesn't mean they don't produce IgG here. They produce IgG here as well. But mainly the main product is IgE. And if, for example, if TGF-beta or BAFF, they produce IgA. So based on the antigen and the type of antigen, T cell provides different cytokine in the germinal center, and in the germinal center, isotype switching happens. So they activated first, they produce mainly IgM, first this one, the short-term plasma cells, and then when they become activated again, they produce different immunoglobulin based on what kind of cytokine how they do it. Based on the cytokine. So how they do that? I hope you can remember this one. No, because you don't read the previous lectures. No, it is the same picture from the B cell maturation. In the B cell maturation, it is the picture I show you. I told you it is the variable region. The variable region is based on the rearrangement, couple of genes, V gene, D gene, and G gene. It is here. And these combination form the BCR. You remember? And when these form produce, they bind with one of these genes. If they bind with this one, usually they bind with this one. That's why the surface IgM comes. The surface IgM comes because they bind with the CMO, this one. But by alternative splicing, we say they keep the variable region but change the constant region, they produce IgD. That's why the mature B cell have IgM, IgD because they bind with these two. Yeah, we mentioned that in maturation. No? You didn't read the B cell maturation talk. Okay, in the B cell maturation talk, we mentioned that one of the V gene bind with the D gene and bind with the G gene. And this rearrangement and combination form the variable region of the antibody, surface immunoglobulin, okay? When the combination finished, directly they bind to the CMO. That's why the IgM expressed on the B cell first. You remember, immature B cell express IgM. It's the reason why. But after that, when they become maturated, fully maturated, what happened? There is a process, they call it alternative splicing. So what happened? They keep this rearrangement and delete this one and bind with this one. That is the IgD. That's why the mature B cells, we mentioned, they express IgM and IgD. This is the reason. So the naive B cell form have a IgM and IgD. That's why when they become activated here, they mainly produce IgM because they don't have anything else. But when the cytokine produced in the germinal center, what they do, they cut this section. They cut it and link this one with the variable. So what is that? That's why they have IgG1. You understand? And the same thing, if it is the IL-4, they cut this part and bind the variable region with the C epsilon, which produce the IgE. So based on what kind of cytokine it has, they cut the FSC fraction gene, they bind it to proper gene, and they produce proper antibody. Based on the type of the cytokine they produce by T cell in germinal center. So we mentioned after maturation, those who doesn't have antigen binding, they will die, and just the only high affinity cells will survive. The reason they survive because they receive a signal by the what?
Abbreviation. They reduce by antigen and T.
Abbreviation. Where is abbreviation?
T. Yeah. High affinity cells.
Yeah, the high affinity cells produce. For production of the high affinity, we need to cells to bind to the T cell and B cell. If they don't have a receptor, they will die. For production of the high affinity B cells, so when they bind with the antigen, they give anti-apoptotic signal. So for production of the high affinity B cells, I told you they need two signals. One from the dendritic cells which present the antigen. What they do, they give them anti-apoptotic signal. Means the BCL2 expressed in the end. After signaling, I told you, it depends what signal they have, the outcome is different. So in this interaction between the surface immunoglobulin antigen, they produce BCL2. And when the BCL2 is expressed, is a positive signal. So they can keep the cell alive. If they don't receive this signal, they will die. Why they don't receive this signal? Because they cannot bind with the dendritic cell. So those who doesn't bind with the dendritic cell, they cannot receive this signal and they will die. And those who bind, they receive this signal. And also there is another signal which comes from the T cells. Yeah.
Doctor, in the previous slide, the last sentence, B-cell endocytosis.
Which one?
This is a presentation of the antigen.
Yeah, it is actually summary of the previous list. I just put the summary here. I repeat it. It's the same.
TFH for the T cells.
Follicular helper T cell, which is only are in the follicular part of the gland. So this is the difference between. It is a difference, please, because I am tired, to be honest. I shouted all the time. So just if you make a noise, nobody can hear from the back. So to help your friend, just become quiet. Not me, to help your friend, because they are there, they have to hear. And so it is a difference between protein and non-protein antigen. Protein antigen is T-dependent, thymus dependent, T-cell dependent, and the non-protein is T-independent. So the difference is the type of the antibody they produce. They produce all the antibody, mainly they produce IgM, and there is no memory cells here, and we have memory cells here, and also the secondary response, you don't have the secondary response. Why? Because you don't have any memory cells. So second response, you don't have in T-dependent. The marginal B cells and also B1 cells, they bind with the polysaccharide. They produce short-lived plasma cells. As we mentioned, they produce just IgM, no memory cells. But what of the problem it is facing with the children? Because children infected with the capsulated bacteria very much, and most of the fertility actually, in the third world is the infection with capsulated bacteria. That's why the scientist was thinking how they can make these antigen to be able to produce the memory cells. What they do, they join the protein to those polysaccharides on the streptococcus, for example. So it is the hapten now. Say they join the protein to the polysaccharide and use that as a vaccine. When they inject this one to the children, they produce long-term plasma cells, and they produce mainly IgG and IgM, and also they produce memory cells. That's why they call it conjugated vaccine. So by joining the protein to the polysaccharide, you can make a vaccine to be able to produce memory cells, which in the normal condition, polysaccharide cannot make it. So by this way, they make it. So in the T-independent response, because we mentioned in protein antigen, we mentioned that the B cell need at least 2-3 signals, and the protein antigen can give them just one signal, and it is not enough. But in the polysaccharide case, it is polyvalent. They have different epitopes. So they activate the B cells in 3 different parts. So the polysaccharide itself can activate the B cells without the need of the T cells. It's the reason, because the polysaccharide is a big molecule and can activate the B cells in 3 different sites. So, and that's why they can activate the B cells. And sometimes they produce a little bit IgG and IgA, but mainly they produce IgM. The reason they can produce the IgG and IgA, because some of the cytokine coming from the non-T cells. In the protein section, they come from the T cells in the large amount, because the T cell can produce it. Since there is not enough cytokine, that's why they don't produce too much IgG. What is the importance of T-independent response? The main function is confrontation with the encapsulated bacteria, like streptococcus. It is the only way they can confront with this pathogen, is B cell which B1 cells and also marginal zone B cell. And also they produce natural antibody. Because if the person didn't get infected, there is a lot of antibody in their body. There is a lot of antibody, if even doesn't. Where it's coming from? Is coming from the B1 cell and marginal zone B cell. What is their function? They don't have the specificity. Because if the B cell, marginal zone, if the protein antigen activate the B cell, the antibody is produced just can bind with the antigen that causes it. They have a specificity. But here it doesn't. They bind with different bacteria and help the innate immune system to be functional. And so that means they have a cross reaction. One antibody can bind with different pathogen, but the antibody produced in the when the B cell become activated, one antibody just only can bind with the same antigen they produce. That's why we use this property to diagnose diseases in the lab. So when a patient comes to you, you suspect that they have a brucellosis or typhoid. You say, please do the test for the Vidal test and brucellosis for this one. What we do, if the patient is sick, they produce appropriate antibody. So we take the serum out and confront it with the different antigen, brucella antigen and typhoid antigen. If they bind together, so it means they have that disease. So that's why we recognize it. The specificity in humoral immunity is very important, but in natural antibody does not have that specificity. It's a couple of slides left. Do you want to leave it for the next time? You leave it for next time. Just 2-3 slides, but this is important. That's why I don't want to say that. Okay. So those people who are going to present it, they can come.